All right, let's get started on today's class. We have a quick announcement. So this year we have a lot more students registered for the course. By this time we have a total of 90, which is about 20 more or 25 more than last year. As a result, the lab, the previously registered lab location is not big enough, at least on the Cambridge side. And so we have changed the lab location into Science Center 309. We are trying to find a bigger room to host for a little longer time. The current lab time is only an hour, 15 minutes. We will see whether there's another group coming afterwards and whether we have to move to, say, the Science Center first floor, that food, resting area. But for now, this is the room we can reserve. We'll try to resolve this. Actually, there are 2 days that's different. Today is one. Today you are gonna be in Science Center Hall A. Okay, and then another date is April 9th. And so you will have a new lab location. All right, so now let's continue with lecture. We talked about RNA-seq in the last lecture, and today we're gonna talk about differential gene expression. We mentioned that the sequencing read distribution actually does not follow a normal distribution. Therefore, the standard tests that people are very familiar with, such as t-tests, would not really work in high-throughput sequencing, at least for RNA-seq. However, there are some simple transformation you can do. In fact, when RNA-seq was first available, there are many groups trying this. They look at the data and they see what kind of distribution it follows. And the very simple thing is, sometimes when the data does not follow normal distribution, you can try to take a log, and after the take the log, they see, oh, okay, actually, if you use the log of normalized read count on genes, and they actually follow a normal distribution. And so a question for the audiences here is, how do we know whether something follow a normal distribution or not? So for that, you can test normality by QQ plot. There's a standard R function. Basically, you know that regular normal distribution, how much data needs to be in certain quantile, how much data needs to be in certain quantile, roughly what range, right? And then if you look at the correct or the observed quantile or the observed data range or distribution versus the quantile, if it agrees with the theoretical quantile on this line, that's an indication that you do have a normal distribution. However, if the raw data looks like this, then you know you shouldn't use a normal distribution test. Okay, and so it's been seen that with high throughput sequencing data, if you look at the read count that lands on a gene, and you look at many samples and look at their differences or variations, you can see that after taking the log of the read count, it roughly follows a normal distribution. Therefore, in the early days, there are algorithms that are developed previously for microarrays, because actually for microarray, after we get the gene expression level, it does follow a normal distribution. And so there are algorithms developed such as LIMMA, which is really using the linear model to study differential gene expression. You can also now use it on RNA-seq. And so in LIMMA, the model is like this. You believe the expression of a particular gene J in condition I in the sample K is equal to the mu. So basically here every gene has a separate ID, and here is gene I. K is a specific sample, and the mu is the mean expression of this gene over all the samples. You can imagine after you get the expression level quantification, you take the log, you get a matrix. The rows are usually the different genes, and the columns are the different samples. And so you can look at each gene and you calculate the mean of this gene over all the samples. So that's kind of roughly how much this gene is expressed. And alpha is the deviation of the mean from this. This is a particular condition. Okay, so I is say a drug treatment, a disease condition or whatever. And so you assume that there is potential deviation of some treatment on effect on this gene, and that would deviate from the overall mean. But if you look at all the different conditions together, the sum of that difference should sum up to 0. That's why you have the mean, right? All the different samples are kind of normalized. Then when we are trying to compare the samples in condition 1 with the samples in condition 2 on this particular gene, what we are really trying to ask is this conditional effect on gene J compared to the control on this gene J, is that effect equal to 0? Is it equal to 0, this is what we call null hypothesis. We don't reject the null hypothesis. We'll say this gene between the treatment and control are not differentially expressed. But if you believe that they are different, then this will not be 0, and you will reject null hypothesis and call this gene as differential. And since if you take a log of the read count, the log, it follows a log normal. You can use a standard t-test or some modified t-test to do this. And the LIMMA algorithm actually does this. You can use this, basically, you provide a gene expression matrix, which has the samples in columns and the genes in rows. And then you also need to specify the design matrix, which says, for example, in this case, the first 3 samples belong to a treatment 1 condition, the next 3 samples belong to a treatment 2 condition, and the last 3 samples belong to the control condition. And then you also need to have a contrast matrix, which specifies which type of comparisons you want to do. For example, the first type of comparison I want is to look at the treatment 1 compared to a control. And in this case, the treatment 1 will be 1, the control will be -1. And so the algorithm will already know that you're going to use these 3 samples to compare with these 3 controls. And then if you want to say, okay, I want to do another comparison, which is a treatment number 2 compare with the control sample, then treatment number 2 will be 1, the control will be -1. And so basically in this contrast matrix, you can specify what type of comparisons you are interested in getting. And so basically, the algorithm will use this type of generalized linear model to estimate across all the samples, what is the mean, what is the effect on each treatment, and whether there is a significant difference between one condition and another condition. And so this works to a certain degree, except that log normal is kind of an overcorrection of your data distribution. And so if you do log normal, yes, it will work to a certain degree, but it's less sensitive in detecting the truly differential genes because you're shrinking this data distribution too much. And so later on, a more sensitive method is modeling the sequence read count distribution by negative binomial distribution. We briefly discussed this. Negative binomial distribution is different from Poisson distribution in that a standard Poisson distribution, the mean and the variance is the same. And so the black line would indicate what that would look like, which is, as we mentioned, for example, you can ask within an interval, if it's completely random, what's the chance that if there's a small probability, there's an event that's going to happen in every small interval. You're asking if we look at a bigger interval. So supposedly you say the probability that the ER will see one patient in 1 hour is a tiny probability. What's the chance the ER will see X number of patients in 3 hours or 5 hours? So this is the same. You are asking if the probability of you seeing one read in every 100 base pair, it's a small probability because the genome is huge and you're looking at one read that lands in that 100 base pair. It's a small probability. Then you ask if my transcript is say 1 KB long, what is the likelihood something lands there? For Poisson distribution, the mean and the variance would be the same, but what people observe that is in real sequencing, there are different type of other biases. And so the end result is that the variations usually is bigger than the mean. And so the negative binomial distribution really capture this. So the strict definition of negative binomial distribution is the number of success before the first R failure if the probability of success is P. And then if you really in this interview, you observed X number of success, what is this probability? There are some standard equations you can get to this. And this is an example of some of these negative binomial distributions with different R and Ps. And you can see here, this data spread is quite different from normal, especially, this kind of look like normal, except that the spread is much bigger. This is an indication of this over dispersion, okay? And so question is, how do we really capture this over dispersion? In any type of, I think most of the time when we are dealing with gene expression, modeling variance could be tricky because in day-to-day gene expression experiment, very often you just treat one drug, you get 2 to 3 replicate, you are trying to estimate the variance in the data. This could be tricky. Normally if you don't have 25 or 100 numbers to estimate the variance, you could be quite off. And so in microarray or RNA-seq experiment, sometimes you only have 2 to 3 replicates in each condition. So how do you really look at the variance? So we use this example to demonstrate. In this case, we don't have even a single don't even have 2 replicates. It's a single replicate. Supposedly you have condition 1 replicate, and condition 2 in only 1 replicate. You just look at some, say, read count, normalized read count. You can see, say, a pair of experiment or one experiment, you might get data like this. In another experiment, you might get something like this. It would mean from here that the second experiment, whatever treatment you have, is perturbing the original condition much less, right? And so even if you have a single replicate and we are looking at supposedly a gene with this level of differential expression between the 2 conditions, even though we don't have replicate, I think you can already see that this gene in the first experiment, you probably don't have very high confidence that it is really differentially expressed, right? Because the noise in that first experiment is pretty high. Whereas this gene in the second experiment, you are much more confident that it is a differentially expressed gene. And so this demonstrates the power of looking at many other genes to estimate the rough noise in the data, even if you don't have replicate. And so that's what people are trying to do when you look at RNA-seq experiments with 2 to 3 replicate, is by trying to look at the noise in all of the genes together. Okay, so how do you do this? We mentioned that, so here you can see each dot is 1 gene, and the x-axis measures the expression level of the gene, and the y-axis measures the variance of this gene. And you can see here, it follows this, the noise is higher than the variance is higher than the mean. And so you can imagine, normally we try to model this, if we try to model this as a negative binomial, this Kij is the, sorry, maybe the Ij is slightly different from the previous slide. So this is the raw count for gene I in sample J. I think previously we have I as the sample and J as the gene, but you get this idea. So this is the actual raw count that we see for a sample. It follows a normal distribution. There is a small scaling factor. This scaling factor is dependent on the whole sequencing depth of your library, and also the gene length. As we mentioned, if the gene is very, very long, then you have more reads. But if the gene is short, you have less reads. It may also correct a little bit of the GC content on this gene. And this is the actual expression level you want to really measure, right? roughly like a TPM level. You are trying to see how much this gene is expressed, and then this is the dispersion. So you can imagine we are observing the raw count, and we say, okay, it's the reason we're seeing this much raw count is because we are sequencing this sample at these steps, the gene is of this length, and if this gene is expressed at this level and there's this much noise, that's how much read we're gonna see. And if we were to draw this figure, you will see something like this, and we can actually model this, the theoretical variance of every gene by this equation, which is the mean ij plus the alpha mu squared. So if we don't have the second term, we are saying that the variance is equal to the mu. That's a regular Poisson distribution. But with this additional term, we are trying to capture this over dispersion. And you can see here, if the sample average or mean is higher, then the dispersion is also a little higher, which kind of captured the situation here, okay? So you can see here the dispersion for gene I is associated with its only expression level, because if the expression is here, the variance would be lower, but if the expression is high, the variance will also be higher. But the theoretical variance, which is kind of this blue curve, we estimate this by borrowing information from all the other genes to really get this curve. And so how would that work is in algorithms such as LIMMA and DE-seq2, the actual variance you observe from very limited sample, these are written as these dots outside the line. The red curve in here is similar to the blue curve in here, which is to really capture how much is that alpha value. You know, how much is the correction are you trying to how much are you trying to correct? Basically, when the data is this is the dispersion divided by the mean count. You can see basically when the sample is at the read count is very, very low, the standard error would also be higher. But when the expression level is higher, the standard error would be lower. And so with every gene, you do the estimate, you try to correct it a little bit towards that theoretical line. So here is the red, yeah, you can think about either the red curve in here, sorry, in here, or the blue curve in here. Okay, so we are trying to correct the variance by looking at the observed variance for every gene, but also the theoretical variance for a gene with that roughly that level of gene expression. And so if the variance is too high from that gene, you try to shrink it a little bit. But if the variance is too low, you try to kind of enlarge it a little bit. The level of correction depends on a few different things. One is the actual expression level, because you can see here, depending on the expression level, your correction is different. And also, how much is this gene varying between the different samples in this experiment? And third is, how many replicates do you really have to estimate this variance? Yeah, question?
How does this work if you so like I know like different frequency analysis, sometimes you might want to take a function of the read count, right? So how would it work if you reparameterize whatever?
So in this situation, the DE-seq2 will just, so basically you look at the different algorithms that use different, you know, they're actually, I would say for this course, over the years, we have tried many different algorithms for differential expression. There's DE-seq2, there's EdgeR, people use LIMMA, they use VOOM, and they all have slightly different parameterization in here. And at the end, it's determined that you just use kind of a normalized read count. This normalization is not trying to normalize between a sample that's sequenced at 3 million read versus 300 million reads. That's too much. You only normalize a little bit between, say, 52 million reads versus 55 million reads, just a little bit, right? And then you just look at that normalized read count, then you can estimate this, okay? Yeah, so basically, the red curve is kind of the correction of the variation or the theoretical variance of the genes at a different expression level. And each of the dots is the observed variance that you see at each gene, and you correct it a little bit by still looking at the data you get from each gene, but also looking at all the other genes to correct it a little bit. So to create a more stable variance estimate. Okay, so with that information, for example, if you have the seq for differential expression, you have sample 1 and sample 2, after we shrink the variance, we can then estimate this probability, you know, whether this is using the original negative binomial distribution, and you're asking whether a particular condition, if you know the mean and the variations, what are variance, whether the observed value is significantly different from the mean. Okay, and so DSeq will give you this calculation in terms of after you find the differential expression, how do you visualize it? There are two different ways people look at it. If you present either of these results to in a paper, usually the reviewer will be okay with that. The one way is called a volcano plot. It's kind of like a volcano shooting off lava. On the x-axis is the log fold change. You're asking between the two conditions. Fold change is just, you know, how many reads you see in the first, how many reads you see in the second condition, and you take a log of that. And the y-axis is a p-value that's calculated based on the previous negative binomial distribution. And obviously, the most significant gene are going to be this blue dot on the left upper corner versus and the upregulated genes will be the right upper corner genes. Okay, that's one way of looking at this. Probably a more informative level of differential expression is by looking at something called the MA plot. So in the MA plot, the M is the log ratio or log fold change between the two conditions, and that's on the y-axis. This is similar to the X on the volcano plot. You can see that's kind of similar. Whereas the x-axis in here is the mean or the average expression level of the two, taken at very often taken at a log, so it's kind of a geometric mean. And so every dot is one gene. And so you are asking if the x-axis is the average expression and the y-axis is the log fold change, you are trying to ask if we look at genes at the different expression levels, which genes are different. And the red dots are all of those ones that are considered to be significantly different. So this way you can at least, you can also figure out, you know, roughly how much is the noise and how many genes are you calling as different and how confident you are about those differential genes. And so to give you an example, if we were to just directly use the mean and the variance that we observed on each gene to calculate the differential expression, on the left figure, this is what we get. You can see here, the x-axis is kind of each dot give you a rough idea how much this gene is expressed in general, and the y-axis give you kind of a log fold change. You see that there are a lot of genes that are called as differential expression with huge fold change. This is just because when the numbers are small, you can easily get a big fold change, right? But then you are not that confident about those genes, and it's better not to really call them. These genes actually probably are truly differentially expressed. And so with the DSeq way of normalizing or stabilizing the variance based on this theoretical variance, by borrowing information from all the other genes, you try to, you know, if any of the variants are too far off, you try to move it towards the center a little bit. You can see here, then the low count genes really, you shrink their variance or you stabilize their variance, then these genes are really not called as differentially expressed in terms of fold change, whereas the higher expressed genes will look quite similar from before. This would mean that you will be able to really know which genes are truly different and which genes are noise. Whereas from here, you can see if you do some cutoff, you will be calling a lot of fake differentially expressed genes. Okay, so this variance stabilization is a very good way to increase your signal to noise, mostly to decrease the noise so that you will be able to have better sensitivity to call differential expression. Okay, questions?